Have you ever wondered, what is the main building blocks of cell membranes? Like you see here, there are walls or a barrier that actually separate one cells from another. Well, that's actually a membrane that encloses the cells, separate cells from its surrounding called plasma membrane. So today, my friends and I will explain to you the structures and functions of each component in plasma membrane. Let us explore it together.
Okay, so the structure you see here is actually the structure of plasma membrane based on the fluid mosaic model, proposed by Singer and Nicholson in 1972. The membranes are composed of two layers of phospholipids. Phospholipid is an amphipathic molecule containing both hydrophobic and hydrophilic regions. A phospholipid is composed of a polar head, which is a hydrophilic region. It's also composed of two nonpolar hydrocarbon tails, which is a hydrophobic region. The hydrophobic tail of phospholipids are facing inwards and no affinity towards water, meanwhile the hydrophilic head of phospholipids are facing outwards, which is outside the cell and cytoplasm and has affinity towards water. So, here's the function of phospholipids. Type of hydrocarbon tails in phospholipids affects the fluidity of the plasma membrane. Also, kink prevent packing and enhance movement in the lateral plane of the membrane.
Next, we're going to explore the purple component you see there. It is actually the globular protein, which play big roles in plasma membrane. There are two types of globular protein. First, it's the integral protein, which is fully or partially embedded within the phospholipid bilayer. There is also the peripheral protein, which loosely bound to the surface of the membrane. So, here are the 6 major functions of membrane proteins. Firstly, it acts as transport protein. It's also involved in enzymatic activity. Third, it involved in signal transduction. And for cell-to-cell recognition. Thus, for intercellular joining. Lastly, it is for attachment site to the cytoskeleton and extracellular matrix.
Next, it's the carbohydrate chain. It's only on the outer surface of the plasma membrane. It functions in cell-to-cell recognition. There are two types of carbohydrate chain. The first one is the glycoprotein, which is the carbohydrate chain covalently bonded to protein. The second one is the glycolipid, which is the carbohydrate chain covalently bonded to phospholipid.
The last component is the cholesterol. It is located between phospholipid molecules in the plasma membrane of animal cells. At relatively warm temperatures, cholesterol reduces membrane fluidity by restraining the movement of phospholipids, helping to stabilize membrane structure. At low temperatures, cholesterol prevents phospholipids from packing too closely, thus maintaining membrane fluidity and preventing the membrane from solidifying. So yay, that's all from us. See you again next time. Bye.